Okay, hello. My name's Tarik Haydar. I'm a professor in the Anatomy Neurobiology department. And I would like to tell you briefly about a project in my lab on neural precursor cells, the stem and progenitor cells of the developing brain, and how they participate in neural circuitry formation. So all of the circuits that we've been talking about today, how do they come to be? And so here is a picture of the cerebral cortex, and immediately you can see how ridiculously complicated it is. It's formed in 6 different layers that you can see sort of laminar structure here. Each one of these layers is produced over time in fetal development. And one of the things that you can probably appreciate is that although many of the cells in a given layer look similar, they're all actually unique and different from one another. And so my lab is trying to understand how this complexity develops and how it is involved in the development of the circuitry. And so as you probably know, the stem cell of the developing brain is called a radioglial cell. And over the course of fetal development, it produces a family of neurons that are situated throughout all of these layers. And over the past 20 years or so, it's become very clear that there are many other progenitor cells, many other types of cells that are themselves derived from these radioglial cells that also produce neurons. And they do this at the same time that the radioglial cells are doing that. And so a couple of years ago, my lab, along with Jenny Lubke's lab, decided to test what the rationale for having all of these different types of progenitor cells is. And we were able to genetically label one type of precursor cell green and another type of precursor cell red, and followed those family members up into the cortex after birth. And so here you can see neurons derived from these 2 different lineages of progenitors nestled together in layer 2, 3. And when we morphologically measured them and electrophysiologically recorded them, it turned out that the red and green cells were substantially different from one another, both in terms of firing properties and morphological complexity, and it had to do with who their parent cells were. And so what we've been doing since then is trying to understand the degree of complexity of these populations. And one of the ways that we've been doing this is with single-cell RNA-seq. And here you can see a map from the mouse fetal brain at a particular embryonic day. And what we have measured are 25 different cell types in this piece of cortex, including excitatory and inhibitory neurons, but also many different flavors of progenitor cells. And so far, we have demonstrated that there are 3 types of these intermediate progenitor cells, 2 types of these radioglial cells, and 2 versions of these other proliferative precursors called basoradioglial cells. And using this gene expression mapping, we have developed now specific tools where we can genetically label each one of these different precursor types and then follow them up into the cortex to determine how they are participating in the circuits that eventually develop. One of the ways that we're doing this is with in utero electroporation, introducing these probes into the stem and progenitor cells in the fetal brain directly, thereby genetically labeling them, allowing us to follow their progeny. And that's it. Thanks.